Good afternoon, everybody. Thank you. We've got a decent crowd here, and between this and our YouTube and Zoom crowds, we do end up putting together a pretty respectful group, typically 50 to 100 people in the audience. It's a real pleasure today to introduce Abby Scott. Now at TESS, as you know, we are primarily terrestrial in nature. We work in forests and grasslands and mountains and freshwater ecosystems and all types of things like that. But occasionally we do actually dip our toes into the ocean. And today we're doing that. It's one of the few talks actually this year that's going to look at marine ecosystems, although in this particular case it's seagrass, which are obviously coastal and near coastal ecosystems, I'm assuming. Anyway, Abby knows a great deal about seagrass. She spent a long time studying it. She's from the UK originally, grew up in Buckinghamshire, which is between London and Oxford, in that general area, nice part of the world. And she studied seagrass at the University of South Hampshire, I think, for her master's degree, and then she came out here. So that's obviously looking at a cool temperate part of the world. And now she's come out here to the tropics at JCU, and where I think there's actually very active groups working on seagrass ecology, coastal ecology here at JCU. So for someone like her, I would think this would be a pretty good place to be. And we're very happy that she's chosen to be with us today. Thank you, Abby, talking about seagrass ecology in the Great Barrier Reef.
Thank you very much. And I'm very excited to be here, dipping our collective toes into the marine environment for a TESS seminar. Before I get started, I just want to acknowledge the traditional owners of the land that we're on today, and also of the lands and waters where I've carried out my research and the research that I'm going to talk to you about today. So today I'm going to talk to you about some grazing research from the Great Barrier Reef. So some of that's going to be my PhD research that I've just finished up at the start of this year. Well, just kind of handed in my PhD at the end of last year, and so I'm officially a doctor. This is my first official seminar as a doctor, I think. So I'll talk to you about some of that work and some of the work that my team's been doing, so the broader work that we've done in this area. But before I do that, I'll give you a little bit of an introduction to seagrasses in case you don't know much about them, maybe having not dipped your toes too much into the marine environment thus far. So seagrasses are an amazing habitat. They are flowering plants that live underwater. So the only flowering plants that we have in the marine environment. And like the grasses that you have on land, seagrasses produce flowers and seeds, but they can also reproduce asexually. And in the Great Barrier Reef, we're fortunate to be in one of the most biodiverse regions for seagrasses in the world. And within the GBR alone, we have 10 species of seagrass, maybe more depending on which taxonomist you talk to, but it's really, really diverse. And it has really important links with other habitats in the reef. So in terms of nutrient cycling and animals that are moving between these habitats, they're all really linked in the coastal environment within the GBR. And they're all really important to each other. Seagrass covers a really large area in the Great Barrier Reef. So we actually have a larger area of seagrass in the GBR than we do of coral. So a petition to rename the Great Barrier Reef the Great Barrier Seagrass Meadow hasn't taken off yet, but I have not given up. It's really important in terms of the ecosystem services that it provides. So it acts as a home for fish and a really great fishing ground. Seagrasses can trap sediments to help protect our coastlines from flooding and store carbon to help with the fight against climate change. They're also a really important food source for a range of herbivores. So that's what I'm going to talk to you about today. But before I get too much into that, a little bit of background in terms of herbivory. Now, as I'm sure you guys all know, being terrestrial ecologists, we've known a lot about how grazing operates in terrestrial grasslands for a while now. Even this seminal paper in 1984 had a lot of detail in terms of exactly how herbivores were using the grasslands that they graze in. And since then, things have really evolved and ramped up. And there's been studies all over the world looking at how grazers in terrestrial habitats can interact with their environment and impact the functions in these ecosystems. Even with new technology, we're looking at on a more bite-by-bite basis how these grazers are grazing, essentially. But in the marine environment or in seagrasses, not so much. So in 1984, the same year as that seminal paper, we had a review about the role of larger herbivores in seagrass communities. where it was stated that direct grazing on seagrasses has been considered relatively unimportant. And it's an elusive question as to why there is no grazing on seagrasses, suggesting perhaps that they're not so palatable. Now, to be fair to this paper, this is all in the introduction, and it does go on to then present some of the evidence that grazing in seagrasses is actually important. But just to say that in a time when we were really getting to understand the detail of how grazers operate in terrestrial grasslands, in seagrasses, we were still debating whether there was any grazing taking place and arguing that perhaps there was. Now, with the marine environment, it is often out of sight, out of mind, and people kind of forget about it. But I think doing ecology underwater comes with its own set of challenges. And I can imagine in 1984, these were much bigger challenges than this suite of challenges that I faced just during my PhD, in terms of having equipment run over, boats break down, losing buoys and having to go into the mudflats for them. So it is a challenging environment to work in. And I suppose that's part of the reason that we're a little bit behind in terms of understanding what's going on in seagrasses. But we have come a long way. And we're starting to understand that grazing in seagrass meadows is really important across the world, from the tropics even to temperate environments. And there's lots of grazer groups that are operating. And we're understanding more how this works all across the world in different seagrass ecosystems, and even how the top-down impact of predation can impact grazers and outcomes for seagrass meadows. We're starting to see that all of these different grazer groups can have an impact. So, for example, if you exclude fish in the Mediterranean, that can have a really big impact on the seagrass meadow. So you can just see that they really are important grazers in this habitat. And sea urchins can overgraze seagrass meadows, so we've been starting to document that. And even the smaller grazers can have a big impact on seagrasses. So we're starting to really see that all of these are important in this environment, and that seagrasses aren't this strange grass that's not being utilized by the animals that are there. So what about in the Great Barrier Reef? Well, we know that there are a lot of herbivores who might be grazing the seagrass here. And so I'll introduce you to them. First up, we have the megaherbivores. So here we've got the green turtles and dugong. And these can eat quite a lot of seagrass. So in the case of dugong, who rely almost entirely on seagrass, they can consume up to 40 kilograms of seagrass a day. And green turtles can switch their diet, but they can consume up to 5 kilograms of seagrass a day. Then we've got the macro herbivores, so the fish and sea urchins. So for example, parrotfish and rabbitfish. These guys will live in the seagrass meadow and feed on the seagrass blades. And mesoherbivores, so these are the small invertebrates that live on the seagrass blades and primarily consume the epiphytes that are growing on those blades. So we know that we've got this diverse community of both the herbivores and a really diverse community of seagrasses in the GBR, but we actually don't know much about the impact that these herbivores are having, or we didn't know much until we started setting up some of these experiments I'm going to tell you about. And it's kind of interesting because the GBR is a really interesting environment to study these interactions, because we have got that really big population of large herbivores, and we have got these large areas of diverse seagrass. So it's kind of these conditions under which seagrasses would have evolved to cope with herbivory. And they have evolved plenty of adaptations to be able to cope with being eaten by large grazers. In fact, a more diverse and abundant grazer community would have been grazing on these grasses in the past. So they're pretty well adapted to cope with it. And the GBR is kind of a small example of how that might have happened. So although we don't know how herbivores, or we didn't know how herbivores are structuring GBR seagrasses, across the world, we've seen there can be a variety of impacts. So herbivores can shift the community composition in a seagrass meadow. So by creating a disturbance in that habitat, they're favoring the faster growing species, and there can be a shift towards a pioneer community, which can potentially increase the diversity as well. They're consuming epiphytes. So like I said, the mesograzers are directly grazing on the epiphytes, but even the other herbivores will be consuming the older leaves that are covered in epiphytes, which is beneficial to the seagrass because those epiphytes will cause shading. They're consuming seagrass biomass, obviously, they're grazers. But it depends on the way that they're grazing in terms of what they're consuming. So you can see here, this is the feeding trails left behind when a dugong's been feeding through a seagrass meadow. So you can see it kind of looks like a vacuum cleaner's been through that, and they're taking both the above and below ground parts of the plant. They can also cause changes in shoot density and height. So if you think about the way a turtle might go through a seagrass meadow, chopping off the tops of the blades, they're decreasing the shoot height in that meadow. And this all has impacts on those ecosystem services that the seagrass meadow provides. So if we've got the seagrass meadow here, and for example, this area is really heavily grazed, that's going to cause those changes in structure that I was talking about. So shorter seagrass, less biomass, perhaps fewer shoots as well. So less structurally complex, which means that area might provide a different suite of ecosystem services. So for example, this structurally complex habitat might be a better habitat for fish, so a better fishing ground. It might store carbon more effectively because it's not being disturbed by grazing. The sediments aren't being disturbed. And because those longer blades are better able to trap sediments, it might protect coastlines from flooding more efficiently. Whereas this other habitat that's being grazed, it's still providing ecosystem services, but just slightly differently. So it's obviously acting as a really good feeding ground for turtles, because that's where they're eating. And if seagrass productivity were to increase in response to grazing, which has been documented, then it might be better at cycling nutrients. And also, it could be a better spot for divers or tourists, because we know that tourists love turtles. Well, I mean, who doesn't? But it's a really good place to go and visit when you know you've got a turtle in a seagrass meadow. And so all of this we kind of put into this conceptual model where you've got at one end a seagrass dominated state with fewer herbivores, more structural complexity, and at the other end dominated by megaherbivores. And so before we started all of these studies, we kind of looked at how the ecosystem services might vary and how the system might operate under these different states to kind of conceptualize what might be happening. And then we went out to do experiments to have a look and looked at the management implications for this as well. Because the way that we monitor seagrasses in the Great Barrier Reef and worldwide as well is by looking at the above ground characteristics of the seagrass. So here in the Great Barrier Reef, we'll go over a seagrass meadow, either in a helicopter or snorkeling or with a drop-down camera, and we'll look at how much biomass is there. And we'll combine this with other characteristics of the meadow, so the species that are there, how much seagrass, and we'll give the meadow a score. So if you were looking at this piece of seagrass, you might think, well, it doesn't look very good, there's not much biomass here, it's in a poor condition. But perhaps as soon as you were to walk away from that meadow or swim away, something else is coming along that's going to be consuming that seagrass productivity. So perhaps what you're actually looking at is a very productive seagrass meadow, but also with some very hungry herbivores who are consuming that seagrass productivity. And if you could understand the seagrass herbivore system as a whole, you might be able to give that seagrass meadow a different rating and understand that it's actually a system that's more in balance and it's in pretty good condition. And this is really important for, like I said, the whole GBR and wet tropics, especially in these areas where we've got large amounts of megaherbivore grazing in particular. So I'm going to talk you through some of the ways we've been looking into this in the Great Barrier Reef. So I'll start with one of my PhD studies where I tried to look at the herbivore community as a whole, so all of those groups of herbivores and how they're impacting seagrass. So I carried out an experiment down in Gladstone, which is in the southern end of the Great Barrier Reef. It's a really industrial location, but there's large areas of intertidal seagrass meadows, and these are grazed on by a range of herbivores, but particularly there's large populations of green turtles and dugongs here as well. So an interesting spot to look at seagrasses. And so I set up an experiment in South Trees, one of the intertidal meadows here. And to understand the impact of the grazers, I used exclusion treatments, which is a common thing that's done in terrestrial environments as well. So to keep out the megaherbivores, I used steel exclusion cages, 2 meters by 2 meters and 0.5 meter high. To keep out the smaller herbivores, so the fish and the urchins as well, I used modified crab pots with a 1 millimeter mesh. And then to keep out or deter the mesograzers, I used carbaryl deterrent blocks. So those were infused in slow release plaster. So like I said, I wanted to understand the interactions between herbivore groups. So I also had to have some treatments combined to exclude multiple groups. So I had my 3 key treatments that I just showed you. I had some control plots pegged out in the meadow. I had a carbaryl block inside a megaherbivore cage. So that only allowed the fish and urchins to access those plots. I had this plot with a carbaryl block inside a macro herbivore cage, which excluded all of the herbivores. And then I had a couple of procedural controls just to account for the presence of those things in the meadow. So all 8 of these treatments stayed out in the seagrass meadow for 3 months. And this is how the seagrass meadow looked. So I had these divided up into 3 sites, which were a couple of 100 meters apart. And at each site, I had 3 replicates of each of those treatments randomly allocated to a grid. So again, these all stayed out in the meadow for 3 months. After 3 months, when we went to take the site down and take those final samples, we could see that it looked like there was a difference inside and outside of those cages. And you could see that the seagrass was looking longer and more dense. And also in image D there, you can see that there was a dugong feeding trail right next to our site. Unfortunately, not straight through it. That would have been really cool. But we could see that the dugongs were feeding in this area. And we saw a lot of green turtles as well while we were waiting for the tide to go out or come back in. So there was definitely a lot of grazing happening here. So how does that look when we actually look at the data? Well, if we look at the change in above ground biomass and change in shoot height throughout the experiment, so there wasn't any differences between those sites, so the same response across all 3 sites. And you can see that in the plots where I've excluded the herbivores, we've got longer seagrass, so a bigger change in above ground biomass than where the green turtles were grazing. And you can see that if you exclude all of the herbivores, so both the turtles and the fish, you don't get any additional impact. So it was really the turtles and the dugongs that were driving this limited change in above ground biomass within the plots that they were grazing in. You can see there was a bit of growth. It was a seagrass growing season, but much less than in those plots that were excluded. So those megaherbivores are really driving those changes. We did also look at below ground biomass at the end of the experiment, but we didn't see any changes, any differences in those treatments, which isn't unexpected because we didn't see a lot of dugong feeding trails or digging right through the site. So no differences there, just in the above ground where the turtles were grazing. Now, I mentioned that I looked at epiphytes as well. So we had carbaryl blocks in this meadow, and they should deter the mesoherbivores, and they did to a degree, but the mesoherbivore abundance was really low. So we didn't see much of a change in epiphytes. So this interaction in this seagrass meadow isn't very important, but we've got very low populations of mesoherbivores and epiphytes. So I'll talk a bit more about mesoherbivores in a second, but here in Gladstone, the main things that we saw was a big impact of megaherbivory. That had the largest impact on the seagrass structure. No detectable impact of the other herbivore groups, and unfortunately no interactions, even though that would have been really cool. The key thing that we picked up was just those megaherbivores having an impact, which they're pretty big and pretty hungry, so isn't too much of a surprise, I suppose. Perhaps this could be because megaherbivore grazing is maintaining this meadow in a lower biomass state in general, so it's perhaps a little bit of a less favourable habitat for those other herbivores, but it's kind of hard to say. It is also an intertidal environment, so it could be to do with that. But don't write off the little guys. So if we're talking about those fish and urchins and even the mesograzers, it doesn't mean that they're not important in the whole GBR, and I'm going to tell you why. So we also did a lot of experiments at Green Island, just off of the coast of Cairns. I don't know if anyone's been there, but it's surrounded by subtidal and intertidal seagrass meadows, really, really diverse, really impressive seagrass meadows. You should definitely go and visit them. And really important for tourists, and also lots of green turtles there. So a good place to study grazing. So I'm going to talk about some work that an honours student in the Seagrass Ecology Lab, Luke Hoffman, did looking at Mesoherbivory in the seagrass at Green Island. So he also used carbaryl blocks, deployed them around Green Island. So usually when you deploy carbaryl blocks in other locations, you're excluding those mesoherbivores, and you'll see a really big increase in epiphytes on the seagrass because that's what's controlling the epiphytes. And once you get rid of the herbivores, nothing's eating them. So you get a big overgrowth, up to 20-fold in temperate environments. It's a really, really big impact. So that's the kind of thing you predict might happen. And Luke did find a lot of invertebrates that were in the seagrass meadow, so he spent a lot of time trying to identify all of these. And what he did find is when we excluded the mesoherbivores, there was an 89% increase in epiphyte load on the seagrass. But epiphyte load at Green Island was relatively low, so it wasn't a huge increase, and it was nothing like the temperate environments. But you can see that when you do look closely at it, there can be some important interactions within that mesoherbivore space. So it's something that we don't know much about in the Great Barrier Reef, and we're starting to just really uncover that. So we definitely need to know more, but it indicates that it can be important in some places. Now, in terms of macroherbivory, or the fish and urchins, I had some experiments looking at the impact of these. So I had exclusion cages out in the meadows around Green Island for 6 weeks. These needed to be cleaned multiple times a week. So cue a crazy looking snorkeler with a broom, very amusing for tourists, but quite effective at cleaning algae off of cages, and quite important. So I had those cages out at 3 sites in the meadow for 6 weeks. Then also did some seagrass tethering. So every 2 months would collect seagrass from the meadow, again at these same 3 sites, and deploy blades of seagrass that didn't have any bite marks, measure the area before, leave them in the meadow for 4 days, and then measure how much they'd been grazed to really look at the rate of herbivory. And I also had some cameras in the meadow to try and catch the herbivores in action. So I had 2 species that I used for the tethering. And you can see here the amount of seagrass lost per tether at those 3 sites throughout the year. And really, it's quite variable over space. So these sites were just all around the island, so not too far apart, but you can see that the consumption is quite different at each site, and it changes throughout the year. So it's really variable. And in the wet season, up to 25% of seagrass productivity was lost at site 1, up to 30% in the dry. At site 2, it was much less, up to 30% in the dry season, but as you can see, it was pretty low overall. And up to 25% of seagrass productivity at site 3 was lost for this species. And if we look at the other species, you can see similar amounts of productivity loss, so up to 23%, up to 13% at site 2 in the dry season, and the highest number was up to 44% at site 3 for Thalassia hemprichii, this other species. So again, you can see here that herbivory by these macro grazers is not consuming huge amounts of seagrass productivity. And in other locations, they've documented more than 100% of seagrass productivity being consumed by herbivores. But up to 44% at some times of year, it still can be a significant pressure on this seagrass. And it's definitely an important interaction within that meadow. So if we look at the total bite marks that I saw on the blades, again, you can see a similar pattern in terms of it's variable across the sites and across the year, but I just wanted to show you the kind of numbers I was seeing for each different thing and what these bite marks actually look like. So again, quite low numbers of urchin shreds, but this is what it looks like when a sea urchin consumes a seagrass leaf. It just kind of goes and shreds the edge of the leaf off. Turtle bites. So in the previous graph, I didn't include loss due to turtles because I was primarily focusing on looking at macro grazers, but here I did count the number of bites because you can see when a turtle's consumed the seagrass that's in your tether, because it's kind of sticking out of the rope and there's just a tiny, tiny amount left and it's cleanly bitten off. And if you don't believe me that the turtles were coming near to my tethers, I thought I'd just provide you with a bit of evidence that they were actually quite interested or quite not bothered by the presence of bits of rope and flagging tape within their meadow, and GoPros, really not too phased. So I also had this number of small fish bites, so that was less than 5 millimeters. And again, you can see quite variable, a bit more important at site 3. And those larger fish bites, again, more important at site 1, but we did see quite a few of these throughout the experiment. So it's variable over space and time, but there's definitely more to be found out about this. And it didn't have an impact on meadow structure. So you can just see here the above ground biomass and shoot density in the control plots and the cages. And I also had the procedural controls out again here. No differences, no impact on meadow structure, which isn't surprising, really, given the amount of seagrass that's here at Green Island. It's a really dense meadow and they're not biting off the whole shoot unless they're turtles. So it's happening and we're seeing those bite marks and it is impacting productivity, but it's not significant in terms of structuring the meadow. Now, was I successful in finding herbivores on my many, many hours of watching GoPro footage? No. There was a lot of bite marks, as you could see from most of my field trips. And I saw some really cool stuff within the seagrass meadow. I saw some of the herbivorous fish, really small ones, hanging out, but I didn't see anything eating the seagrass. So perhaps these grazers were grazing at night, or perhaps they were coming in shoals that I just missed. It's a really interesting question that I really want to know the answer to, because there's obviously quite a bit of herbivory happening at times around Green Island, and I put a lot of cameras out but didn't catch any herbivores feeding. But anyway, there's plenty of cool stuff to see in the meadow, so it's not bad footage to watch. We did see some herbivores when we were swimming around. So for example, rabbitfish and sea urchins in the meadow. So we did see evidence that they were there along with the bite marks, but none of them feeding on film, unfortunately. We'll have to wait for that another day, I guess. So in terms of the macro and mesoherbivores within the GBR, mesoherbivores can impact the epiphytes on the blades, but they're not having such a big impact as we're seeing in temperate meadows. Macroherbivore feeding, it's not impacting the structure of the seagrass meadow, but it is happening, and we're seeing that they are consuming up to 44% of productivity at Green Island. It's just very variable over space and time around Green Island. So again, that's kind of really just scratching the surface, and we're just starting to find out how that's operating, just that one seagrass meadow. But it's interesting to note that these interactions can be important here. But really, as we've seen with that first experiment, it's all about how the megaherbivores are feeding. And so, as I mentioned, my PhD was part of a larger study into megaherbivory, which had exclusion cages out throughout the GBR. So all these sites you can see up here. And these were the same exclusion cages that I used in the previous study, and they were out for a year plus, depending on the site. And so we had some really interesting results as part of this study, looking on a GBR-wide scale at what the impact of herbivory is for the first time, really. So up in the Howick Islands, you can see this is reef-top seagrass, so it's not particularly dense, but you can see inside the cages after exclusion, much more seagrass there. At Lizard Island, the turtles really did want to get into our cages because the seagrass did look that good. So more cages inside there, more seagrass inside those cages there. Again, at Green Island, we can see a big impact, and I'll come back and talk a little bit more about Green Island because we observed some interesting behaviours there. And then Yule Point, just off the coast of Cairns, you often see dugong feeding trails through this meadow. It's, again, an intertidal meadow, but quite a big impact visually at the end of the experiment. So how does all of this look when you look at the data? Oh, sorry, my sites have gotten a bit, my site names are a bit mushed together there, but they're all in the order that they are on the map. And you can see there that if we're looking at the above ground biomass at these sites, there's an impact of putting these cages out after a few months, depending on which site you're at. But you can see at some of these sites where it's highlighted yellow, that's where there's a significant difference. So at all of these, apart from Palaranda and Bowen, we're seeing an impact of herbivory on structuring the seagrass meadow in terms of the above ground biomass. And if we look at the canopy height, we can see that impact a little bit earlier at some of those sites, which is similar to what you observe often in terrestrial environments, where you'll pick up changes in canopy height and the biomass will still stay higher. You can see that for some of these sites, so like Lizard Island Deep and Green Island Reef, or some of the ones where there's smaller seagrass, like the really short species, we're not seeing as many differences. But again, we're picking up big differences in canopy height in terms of excluding these herbivores at a lot of the sites throughout the GBR. And there are differences, so those are differences over space, you know, and most of the sites we're seeing quite a big impact of excluding the megaherbivores, but we do see differences over time as well at the same site. So if you remember the study I spoke about first of all in Gladstone was conducted in 2017, over a 3-month period. At the end of that experiment, we could see a big difference inside and outside of the cages. Well, as part of a slightly different study, I went back in 2018 and put cages out in a very similar spot at South Trees at the exact same months as I did before, and I saw no difference whatsoever inside and outside of the cages. So it's really dynamic in terms of where the herbivores are moving and how the seagrass is responding to herbivory and the other things that are playing into seagrass health. So you can't really just go and say, well, this is a site where it's important, because it's so variable even over time in the exact same place. And the scale of impact that we were looking at most of these meadows, so in the study at Gladstone, I didn't see any differences in those 3 sites that I looked at. And also throughout those studies up and down the GBR, we didn't notice any particular focused grazing. It was the grazers appeared to be going through the meadow as a whole and just feeding all around the meadow. There didn't appear to be any hotspots. But at Green Island, we found something a little bit different and something really, really interesting. So this is back to one of my PhD studies. And again, I had 3 sites set up. So similar to in Gladstone, these were 200 meters apart from each other. And at each site, I had 3 cages and 3 control plots. And so this is what the site looked like when I set up the experiment in August. Really long, lush seagrass inside and outside of the cages at all 3 sites. And we'd go back and check these cages every 2 weeks. And for most of that time, we saw no differences inside and outside. And after 2 months, we went to site 1 and site 2, long, lush seagrass everywhere, which is brilliant, but not really a cool impact of grazing. But we jumped in at site 3, and this is what we saw. So all of a sudden, we'd lost a lot of the seagrass. Now, we had some strong suspicions about what was happening here, and we thought it was due to grazing, but we weren't sure if it could be down to some other impact that we hadn't quite seen or foreseen, because this was a really localized impact just at this one site. So the next week, we went back and we put a GoPro down to try and figure out what was going on, and the guilty party came and started grazing around our cages. So we could see that it was due to turtle herbivory. And what's interesting about this is that as the experiment went on and we kept monitoring it, this grazed patch didn't really get any bigger, but it did get a little bit barer. So it seemed like the turtles were really just focusing in on this one area when they could have fed anywhere in the meadow. There was really long, lush seagrass everywhere. So this bare patch perhaps wasn't the tastiest meal or the easiest that they could just go, but they were really focusing in on it. And we could see this in the above ground seagrass metrics as well. So from the end of the experiment, we took cores, and you can see sites 1 and 2, where we didn't see any grazing, there was no difference in above ground biomass. But site 3, in that control plot where the turtles were, it was much lower above ground biomass than all of the other plots and the cage next to it. And similar pattern in shoot density. The differences weren't significant, but you can see that there is a reduction in the shoot density inside that grazed plot that the turtles formed. And had we kept monitoring that for a little bit longer, we might have seen even more differences there. You can see in this shoot height data throughout the experiment, or canopy height, exactly when the turtles moved in and the kind of impact that they were having at that site. Now, we also looked at below ground seagrass metrics, and you'll remember that at Gladstone, there was no impact on the below ground metrics when there was grazing. But in this graze plot at Green Island, the below ground metrics were also much lower than the above ground metrics. And so we did see the turtles grazing on the rhizomes, and that's really interesting because that hasn't been documented in many cases before. So this paper was from Christian in Indonesia, and they documented turtles digging seagrass with their flippers and pulling the rhizomes out. And we also saw at our site some holes that looked like they'd been dug, but also on the GoPro footage, we could see the turtles really pulling out the whole shoot and the rhizome. And so that's really only one of the second times that that's been documented on film after this paper from 2014. So really interesting to see that the turtles are doing that here. So in this grazed plot that the turtles formed, there was a 60% reduction in above ground biomass, 75% reduction in canopy height, reductions in shoot density, and also a 60% reduction in the below ground biomass. So a really, really big impact on seagrass meadow structure. So why did the turtles feed like this? Well, we thought perhaps it's because there was more biomass here, but we looked at the biomass data and we saw that there wasn't really a big difference, and there was plenty of biomass everywhere. Were they perhaps targeting a certain seagrass species? Well, if we look at the seagrass species at site 3, at the start of the experiment, there was a little bit less Halogila uninervis and a little more Syringodium, but not really a big difference. There was plenty of all of those species available to them. So it doesn't seem like they were targeting a certain species. Perhaps they were looking for nutritional content, as herbivores on land do. And so we looked at the seagrass inside the megaherbivore cages from site 1, which wasn't grazed, and site 3. And we saw that elevated nutrients in almost all of the species at site 3, which was grazed. But it's kind of difficult to say definitively because we did take these samples from inside the cages that were protected from grazing. So it could have been there was some translocation of nutrients into those seagrasses as a response to the grazing that was happening. Unfortunately, we weren't able to sample enough seagrass from the control plots within that grazed plot because there really wasn't much seagrass left by the end of the experiment. So perhaps it could be related to nutritional content, and that's something that would definitely be worthwhile investigating. But also really important to remember that they were excavating those rhizomes and there was impact on the below ground biomass. So seagrass rhizomes are really high in carbohydrates and starches. So perhaps the turtles were feeding like this at Green Island to be able to access those for some kind of a dietary need. It would be really interesting to know, and hopefully we'll be able to find out. Well, is this a repeated behavior? So in other locations, people have documented turtles forming these grazed plots and kind of moving between grazed plots. In the Great Barrier Reef, this is the first time that we've seen any formation of grazed plots by green turtles until we put the cages out as part of the broader ARC study. And so these cages in the Green Island Lagoon were very close to where I had my cages. So I'll show you on a map in a second. And you can see here that after a little while, there was a bit of an impact on the canopy height from grazing, I think just from them browsing in the meadow. But then if you look, there's a huge difference here. And so something has obviously happened, and that's reflected in the above ground biomass as well. Suddenly there's a big difference. And once again, we've seen that there was a grazing plot formed by the turtles, and you can see that there was kind of some, it looked like there might have been a bit of digging going on, a few holes within the meadow. So is it a repeated behavior? Well, the grazed plot that I documented in my PhD was formed in October 2017 at Site 3. And these cages that then went out later were really close to where I had my Site 2. So not too far away in the meadow, but we didn't observe any impact until July 2019. And then all of a sudden, boom, there's another grazed plot. So it looks like this is a repeated behavior at Green Island, but we just don't know whether the turtles are grazing like this and regrazing the nutrient-rich regrowth of the seagrass, which has been documented, or whether they are moving between set grazed plots and letting them grow and then regrazing them. We just really don't know much about the dynamics of what's happening here just yet, but it would be really great to know. So another thing that megaherbivores can do is to transport seeds. So we know that on land that herbivores can ingest seeds from plants and they can transport them, and then they'll come out in their feces at the other end. And so this is some of the work that another PhD student in she's now graduated, Dr. Samantha Toll, carried out while I was also doing my PhD with the Seagrass Ecology Lab. So Sam's research, she went around and picked up poos from the sea and sieved them and looked for seagrass seeds to see if they were viable, because even though we know a lot about how this operates on land, we just didn't know whether this was a process that was operating in the marine environment. And so Sam found out that megaherbivores are transporting seeds in their poo, and they can transport seeds up to 650 kilometers in the Great Barrier Reef. So really important in terms of contributing to the genetic diversity of the seagrass meadows here, and also in terms of recovery and even starting new meadows, perhaps. And so she did that work in 2017, and then she went on to take that a little bit further and look at how the seeds being ingested can impact their germination. So she found that megaherbivores enhance the germination probability of seagrass seeds by 2 to 4 times, and the seeds germinate faster. So when we're talking about herbivory in seagrass meadows and how it can decrease the meadow structure, we have to remember that it's not all bad. You know, these have evolved together, and there are benefits that herbivores can bring to seagrass meadows, such as spreading seagrass seeds and enhancing their germination. So in terms of megaherbivory, what are the conclusions? Well, we can see that it's structuring the meadows within the Great Barrier Reef. Grazing plot formation is taking place within the Great Barrier Reef at Green Island, but there's definitely more questions that we can answer about that. Below ground biomass is consumed within these grazing plots, and the megaherbivores are transporting seagrass seeds and promoting their germination. So in terms of what we thought about the role of herbivory, that direct grazing on seagrasses hasn't been considered relatively important. Well, I think we've moved on from there, and we now know that grazing on seagrasses worldwide and within the GBR is really important. It's a really important process. And the key questions that need to be answered are not why there's relatively few grazers on seagrass meadows. It's more like, what is the impact of grazing and what is the role of herbivory in seagrass losses? So we're seeing around the world some loss of seagrass that's being caused by overgrazing. Now, in the Great Barrier Reef, we seem to have more of a balance in the system that we've got here. We've got top predators controlling our herbivores. We've even got hunting, keeping a bit of a top-down control on those herbivores. And we seem to have a system in most locations that's relatively in balance, but we do need to understand what the role of herbivory is and what a seagrass herbivore system looks like and how we can best manage for that kind of a system. So if we go back to that original diagram that I showed you, we might put Green Island more onto the seagrass-dominated end of this spectrum, whereas we might put Gladstone more on the megaherbivore-dominated end, where the herbivores are reducing the seagrass structure and potentially impacting the ecosystem services that that meadow provides. And since we published that paper, we've had other people have their own take on this concept and kind of change this figure a bit to understand more about how the turtle grazing really is operating. And it's interesting because at Green Island, we could be having that kind of a patch rotational grazing pattern, whereas at Gladstone, it's more of a random constant pressure on the meadow. And throughout the rest of the GBR, perhaps a bit more of a random constant pressure where we haven't seen grazing plots formed. But again, there's still more that we need to know about this. And in terms of management and monitoring, it's really key to understand how the seagrass herbivore system operates together, because you can't really just manage for one or the other when you've got both of these interacting within the Great Barrier Reef. And there are going to be impacts on ecosystem service delivery, and we need to be able to understand more about what those are. You know, we're understanding the differences in seagrass meadow structure, which will impact the ecosystem services, but there's definitely more to know about exactly how that will operate and exactly what your management priorities are when you're managing a seagrass meadow. And we obviously want to be preventing any herbivores contributing to seagrass declines. And like I said, that's not a big issue that we're having in the Great Barrier Reef, but it's something that's happening elsewhere. So it's definitely key to understand the dynamics that are happening here and prevent us reaching any of those kind of tipping points. So in summary, megaherbivores are super important, but the mode of megaherbivory is different between those sites. So we can see different ways that herbivores are grazing here in the Great Barrier Reef. In terms of the fish and the invertebrates, they can be important, but they're not a huge structuring force, especially when you're comparing that with the larger herbivores that are here. And understanding the seagrass herbivore system is really, really key in terms of conserving both the seagrass meadows and the herbivores that rely on them. Thank you very much.